We are ready for the second day. So our first speaker today is Klapper, and he will talk about biofilms and bottles for biofilms.
Thank you. So I apologize that I didn't have an abstract. So I wasn't sure what I should talk about, and by the time I decided, I forgot that I hadn't submitted an abstract. So what I want to do is talk about biofilm models, a little bit about biofilms, and I decided, so that's a big topic, of course, so I decided to focus on physics of these things, which is still a big topic, but we'll see how far we get. But I was entertained enough yesterday that I thought I'd say something about modeling this as well. So this is my favorite modeling quote here. And of course, this is irony. The idea that the writer is trying to get across is of course sort of the opposite. One, in my view, is one wants to throw away as much as you can, but not more than you should, and not try to put everything in and be as accurate as possible. The goal in a lot of kind of models is not necessarily accuracy, but insight in a certain way. So mathematical models, sometimes there are in some cases you really want to make quantitative predictions, but often the idea is to add to qualitative understanding and to see a phenomena that you're interested in and think about what properties of your system cause that phenomena, and you can come up with a hypothesis that way and test it with a model. Or you can have some interesting properties of your system and you wonder what they may do, and you throw them into a model and see what comes out and maybe generate some new hypotheses. And with mathematics you can do this in a relatively clear way by getting rid of a lot of things that may interfere with you and often inexpensive, relatively speaking. So that'll be one of the themes here. Okay, so let me say a little bit about biofilms. I don't really know exactly who's in the audience and what they know. There's probably a lot of people here who know more about biofilms than I do, but there may be some who don't. This is a picture of a chunk of a biofilm. It's actually a microbial mat, so this is quite thick, a centimeter or more, from Yellowstone National Park. Notice the very top layer, I don't know how well you can see it, but it's a little green layer on top of a millimeter or so. That's the most active part of this biofilm. That's photosynthesizing. Okay, so what biofilms are? Well, a loose definition. They're collections of various microorganisms that somehow come together and secrete polymers and form a sort of city. People use the word biofilm in a lot of other ways, which is fine with me, but this is, if there is an accepted view, maybe this would be it. So here are a couple more examples. This is again from Yellowstone. This is quite big, photosynthesizing algal mat, and this is a lab-grown biofilm. I don't know the scale on this, but it's in tens of microns probably. A little bit of natural history. This is the tree of life, and the microbes are most of this, as you know. And the biofilms fit into this basically all the way through here, and they show up in fossil or indirect fossil records close to the beginning of life. This particular picture here is also from Yellowstone National Park. These are Oncophacales streamers. These are chemoautotrophs, and I put them here because these are down on this bottom branch. So even in the bottom branch, at least in modern day, you see biofilms. These are stromatolites from Australia, which are thought to be remnants of things that were around a long time ago, and for which there seem to be fossils as well. So here are SEMs of the EPS matrix, which is a little bit hard to see normally. Of course, This has been prepared, and so whether the matrix really looks like this is another question. But you can see that there's a lot of stuff, a lot of gunk in there. Okay, and here are a couple of cartoons to give you the idea. If you haven't noticed, biofilms are generally not necessarily flat, despite the name. And they contain, maybe microcolonies or something like that, and EPS, and there's flow over the top, and there are boundary layers, and there's streamers that flap around, all sorts of different things that go on. Okay, chemistry is also important, and if there's time, I might talk about that a little bit. These are not biofilms, but these are effects of biofilms, or maybe the things that affect the biofilm. In this case, we have mineralization, basically likely from a biofilm raising the pH, and this is likely from a biofilm lowering the pH and causing demineralization in some way. Oops, I got the wrong caption here, so ignore that. Just a few more unusual biofilms. This is off of a contact lens. Biofilms can grow pretty much anywhere where there's at least a little bit of water and some chemical-free energy, or light-free energy in this case. This is a biofilm, a subaerial biofilm, so it's not actually submerged. Occasionally it gets rained on and there's some humidity around. This is actually the top of the Lincoln Memorial in Washington, D.C. This biofilm came from there. And this dark stuff, well, we don't really know exactly what that is, but there's certainly biofilm growing in this stone, phototrophic biofilm, and this is an example of it. The blue is actually the phototrophs. Those are not cells, they're little colonies of phototrophs, and these green are heterotrophs growing on top. So there's a community going on here. I'm not going to talk about ecology really at all. The red is actually EPS, the polymer stuff here. So this is the top of the biofilm up here, where it looks like a lot more activity going on. Okay, so what goes into a biofilm model? Of course, you have the biology. So a lot of the interest is maybe in some of these things up here, which I won't really talk much about at all. And then there's physics, which is very important here. I want to stress that, and chemistry, chemical physics, if you will. All these things by themselves make things complicated, but in addition, you have spatial non-uniformity, which has an enormously important effect. Adds all sorts of interesting things and also makes things more complicated. So how do you actually put together a model? Well, there's basically two pieces. There's the dissolved components, which I think pretty much everybody does with PDEs, vection diffusion equations. And then there's the particulate components, the non-dissolved components, if you will. And Jan Kraft yesterday talked about using discrete models. I have always used continuum mechanics models. My background is fluids. So I'm only going to talk about this, but just because I'm more comfortable there. Okay, so the first thing I want to talk about really is transport in a biofilm, which is often a fundamental, the fundamental issue in many cases. So this is a picture. I think it's a Pseudomonas aeruginosa biofilm. Which has been stained, and then the active part counterstained, the metabolically active part counterstained, and you can see this layer. This is the top of the biofilm, and the oxygen is up here, but not down there. So there's a very sharp active region here, and below that, things are inactive, and that leads to a lot of the important properties of biofilms. You have the activity going on here, and below you have some protected layer, so it's protected from antimicrobials and other types of things. So this is really a fundamental property of many biofilms. So I wanted also to talk about a chemostat a little bit, and I'll tell you why in a second, but I thought in case you're not sure what it is, I'd show you a picture. I just grabbed this from somewhere. It's not the best picture, but gets the idea. So you can think of a chemostat this way. You have a tank of fluid that's being mixed well, and you have some stuff coming in at a certain rate, and stuff is going out at the same rate. And so what that does is really fix the rate of everything that's going on. So I didn't really appreciate this until relatively recently when an engineer explained to me why this is so interesting. So here are the equations I think we saw yesterday. You have a population density, a substrate density. This is the simplest version. And the population has growth and washout, so the growth looks like this. 1 over d is the rate that this tank, is the time rather, that this tank takes to fill or unfill. And then a similar equation for the substrate. Well, you have an extra term, substrate is coming in from the outside, but then there's usage corresponding to this term, and then washout. This is a little bit like a biofilm. In some ways, it's not a bad place to start thinking about a biofilm, a chemostat. So a biofilm, you have a layer, and typically you have, over the top of it, you have fluid coming, which is supplying you with nutrients at a certain rate. And that's what you get if you live in the biofilm. You have things coming at a rate that's fixed by the outside, and you have to deal with that. So basically, it's not a matter of how fast or slow you grow. You have to grow at that rate, basically, the rate that you can bring things in. That's how a chemostat works, anyways. So one thing I think this is on the next slide. So one thing that's particularly clear about a chemostat is equilibrium. So if you let this thing go for a while and this inflow rate is constant, you can find these equilibrium solutions that look like this, just setting the derivatives to 0. And the important thing is actually this equation here, which says, well, either x is 0, meaning there's no population, it got washed out, or the rate is set by the rate of the chemostat. So in the chemostat, everything goes at the same rate. You don't have the freedom to say, I want things to grow faster or slower. You have to grow at that rate. It's a constraint. So the chemostat population is transport limited. It's how fast you transport things in determines a lot of what goes on. The other thing to think about in an equilibrium like this is, well, so we have growth, but then to balance that, you have to also have some loss. And it's clear what that is in a chemostat. That's the washout piece. So at equilibrium, the loss rate through the washout, I should say washout rate, equals the growth rate. Just as an aside, by the way, it's not difficult actually to make these steady states unstable if you start playing around with this thing. So by adding, even if you keep everything, sort of the boundary conditions constant, just by adding in species, you can get things to go or different substrates, you can get things to go unstable pretty easily. Okay, so let's now think about a biofilm. Biofilm population is often also transport limited. The growth rate is set by how fast you can bring things into the biofilm, how fast you can transport them. You have a constant rate typically, or some set rate anyways, of nutrients and stuff going over the top. There are some differences. Space limitation can be important in a biofilm that's not in the chemostat. And another important one, which I want to stress, is, well, if you want to look for an equilibrium or some version of an equilibrium in a biofilm, then you have to balance growth rate. So somehow, as a biologist, it's very natural to think about how things grow, how you generate new biomaterial, but less natural to think about how you lose this material. But if you want to think about equilibrium, what happens over long times, then you need to know how that works. And at least as far as I know, this is really not well characterized for biofilms. So what you do, if you still may want to know what happens over a long time, so you have to impose some ad hoc mechanism to enforce balance. It's not clear if this is a good thing to do, but you don't have a choice here. And just as an interesting aside again, even fairly, so we talk about steady states for biofilms, even fairly simple 1D biofilm models can be made to oscillate pretty easily. You apply constant, well, I'm not sure how easily, but it can be done. You apply constant boundary conditions basically, and you can force these things to have unstable equilibria and sort of oscillate in between them. This is, I mean, these whether this is corresponds to real oscillation in the environment, I or in true biofilms, I don't know. This is just an issue. Oops. This is just an issue of this artificial loss mechanism, really. So it's a mathematical thing. It's a problem with the model. Okay, so let me get a bit more specific. I want to start with a 1D biofilm setup. So and you have a homogeneous system, and by 1D I mean that things vary in this direction, but not in the horizontal directions only. So you have a biofilm here. You have, say, a well-mixed region with a supply of nutrients or something, and a boundary layer between, possibly. Okay. So here are our equations. This is the equation for a substrate. I only want to talk about substrate for the moment. And this is a reaction diffusion equation, basically. This is the diffusion, this is the reaction, essentially. And you have a Dirichlet condition on top. You have a given amount of nutrients out here somewhere, and no flux condition on the bottom. Well, typically this is a simple thing to do. It can be different in different cases. And then, well, so what goes on? The total activity in the biofilm is proportional to the total yield, generally, and this is maybe something you want to know. And the total activity, you want to take the rate that things are being used times the yield and integrate it over the biofilm. And if you do that, you get this expression just from here. And we can see a couple things from this. One is that if you look at this equation, it tends to have exponentially decreasing solution. So the contribution to this integral tends to be from an area close to the top. That's the active layer. In fact, you can estimate it, the depth of this layer in this way. I won't go into details. And so we reproduce this behavior. So basically, the substrate gets eaten at the top, and nothing gets through. I mean, it's just as simple as that. The other thing is that this guy is really the important thing is C prime at H. D times C prime at H. That's the flux in at the top. That's what controls everything. How much stuff you can push in through the top. That's really one. Everything comes down to one number here. Of course, that's for one substrate. You could have more. For example, you could have a bunch of different electron acceptors. As in this cartoon, you use the best one, and then the next best one, etc. And in that case, you can get a bunch of layers. And just to inform you, here's the type of equations we're looking at generally in three-dimensional. This is our advection diffusion reaction equation. Typically, you can neglect this piece and set it to 0. This thing, there's not much velocity inside here, and you equilibrate quickly compared to the other time scales involved. So typically, it's just this guy equal to 0. Excuse me. Okay, so here's our equation. This is what I wrote down before, and what I said we really only need to know C prime of H. You can actually partially integrate this equation to get an expression for C prime of H, and often you can basically bring it down to this guy. These things are all known in principle. This is the rate of usage of substrate out at the level in the mainstream, and these are just other constants. So you can get a decent estimate. And notice this is basically independent. It's almost independent up to under fairly reasonable assumptions of what your kinetics look like. Just depends on the growth rate at a certain value of substrate up in the mainstream. You really don't get much of a difference as you go from one form of kinetics to another. And the idea is basically everything's happening near the top. So it doesn't really matter what the rate function looks like for values that are not close to what at the top. Not very much, anyways. It's really only a square root of 2 difference. At most, you can bound these things. Okay, I want to quickly look at a different type of biofilm. We'll call them embedded biofilms that come up maybe in host systems. So this is a picture of a chronic wound, and there are a lot of little microbiofilms growing in here. It's thought. Here's some pictures from the literature. These are from chronic wounds samples, and all these little lit up things are microcolonies, if you will. So rather than one biofilm, and they're embedded in tissue, rather than one biofilm, there's lots of these little things. If your eyesight is bad, like mine, and you're sitting in the back of the room, you might not, it just might look like pictures from before. It just all kind of blurs together. Here's another one. This is from a cystic fibrosis lung. We have lots of chunks of biofilm embedded in mucus or whatever. So the question is, for these systems, we know a lot about 1D biofilms and 1D biofilm models. And for these heterogeneous systems, when can we think that they look like 1D biofilms, and when do they not? And how exactly would you do that? So this is just a recent thing that we've been looking at. Here's an experimental system. Brianna Paps is a student running this, and you probably can't see that. I can't even see it from here, but there are a lot of little, so you start at t=0 with a bunch of embedded cells in a gel, and the gel is open here to oxygen, and on these walls it's closed. Actually, the gel goes down farther, but basically the only oxygen way for oxygen in is up here, and then you let this thing grow, and the green is active stuff. And you can see over time, this looks kind of like the picture that we had before, an active layer on top with things below. So basically you might think, well, this is going to act like a 1D biofilm. It's going to block oxygen from getting down here, and all the intuition we have about these things could apply here as well. Okay, so the way we attack this problem is to try to homogenize this. So this is mathematical homogenization, the lab version. So the idea is if you have a heterogeneous material, so you have properties that change in a certain way over the course of the material on a small scale, and you're interested in a larger scale, perhaps you can somehow average it if you do this properly and replace this material by a nice homogeneous one, which on a small scale, of course, you won't get the right answer, but on the large scale, you will. That's the idea of the theory. It's a well-established theory. I should say, even if you can do this, there may be some surprising properties. So you may go from, for example, isotropic properties for each of these phases to anisotropy here. We, in fact, have seen that. There are other things that can happen, too, but we actually have data in real biofilms where that seems to happen. And in particular in diffusivity. And what we see is that the diffusion in the vertical direction is increased at the expense of the diffusion in the horizontal. So the idea being presumably to increase transport rates. That's what's limiting. So the biofilm in some way is deciding to act as a community and change its physical properties. Okay, so the question is when is this valid? When can you homogenize? And the idea is this: pretend that your microbes are smoothed out uniformly. So you go to a nice continuum uniform biofilm. In other words, you want to defocus your microscope like I described before. Now for this uniform thing, approximate an active layer depth. So what would be the active layer depth if you smooth this thing out uniformly? Compare that length to the distance between these colonies. If the colonies are close together, then basically with your smeared out poor vision, you can't see that they're separate colonies. They just look like everything's smoothed out. So in other words, the interesting length scale is the active layer one. If the colonies are closer together than that depth, then you can homogenize. If they're farther apart, then you can't. Okay, so just a little bit more precisely, you have an effective active layer depth, which looks like the same one we had before, except now we include a volume fraction. And then you have a length scale between colonies, and you basically just look at the ratio of these two lengths. And if you get something around 1, then your active layer depth is about the same as your space between colonies. A small indicator indicates that the colonies are close together. You can think of this as a 1D biofilm and just go ahead and use your intuition. If not, you really have some scattered system where you have to take account individually of these things. So here's our model computation. So we're looking at a reaction diffusion equation here for colonies far apart, close together, and in between. So what are you seeing here? This is a layer. Oxygen, say, is 1 or something here and 0 up here. So if there was nothing there, you'd see something like right here. You go linearly, basically, from one color to the other. And basically, that's what happens in this case where there's few colonies. On the other hand, if there's a lot of colonies, if it's like a 1D biofilm, you get an active layer. So basically, all the oxygen is gone by the time you get to here. Okay, so like I said, this is a large indicator and a small indicator. And you might be thinking, well, of course, there's just a few of these things, so they're not going to do anything. But the catch is these are super strong colonies compared to these. So what we did is we took the total strength, if you will, of the colonies here is the same in each of these pictures. So these 3 colonies, or however many there are here, are in strength the same as all these zillions of colonies. Okay, but despite that, and you can see they have quite a local effect, but globally they don't do much. Okay, so numerically this seems to work pretty well. Basically, the idea is you're just comparing the active layer depth or putative active layer depth to the distance between colonies to see if you have something that's like a true biofilm or a bunch of microcolonies. We're working on connecting it to experiment. Here's a comparison, and the important thing now is flux. How much flux do you push through this thing? Lambda over epsilon is this indicator, so somewhere around 1. It depends on how you define epsilon, but roughly around 1. This is the error, so we're comparing the flux through our system with the flux if you had a true one-dimensional system. And they're basically about the same. There's some error that's left over, I think, basically for some numerical artifact reasons. For large value, but small value, it's quite different. Okay, so that's transport. What's transport good for? You want to see growth, right? Everybody wants to see growth. So in that case, you need to go to mechanics. Right. So we ignored that in our model because we didn't need it for transport, but now we do. So we have to put that in. Here's the simplest sort of model you might do. Growth-induced mechanics. So the thing is growing, and space is at a premium. So unlike the chemostat, space matters. So if you grow, you have to push other things out of the way because there's no extra space, maybe. So the idea is you have some substrate, it diffuses in, just like we were talking about, it transports in, it gets eaten in an active layer. But now that causes growth. So there's going to be some expansion here. This pushes outward, it's shoving outward, and so the biofilm has to expand. Okay, so we have to add some mechanics. All right, so here's what we had before. These are our dissolved components. Now we actually have to have a transport equation for the particulates as well, in principle, although in this very simple model, it turns out you don't need it. So now these are, say, the different species or something, and they have advection and possibly diffusion, and then reaction is growth, or some other sort of motion term here. But what we really need is now a force balance, conservation of momentum. This is Newton's law, essentially here. So we're in the home of Newton here, and we can talk about Newton's law. You need this to determine these velocities, in particular the one that goes here. And a lot of things can go into that, and you have a moving interface now as well, which can, in principle, make things very complicated, but you want to be as simple as you can get away with. And so this is maybe the simplest thing you can do. So here's what we already discussed. This is the substrate equation, diffusion reaction equation for the substrate. And then we need a velocity for deformation, and the simplest thing that I could think of anyways is to add a growth-generated pressure. So you're growing and you're pushing out, that causes a pressure, and make the velocity proportional to that. Okay, well, so we've introduced this pressure. We need an equation for that, and the idea is basically you just expand. You put in a divergence of your velocity to account for the growth. So you just every point is pushing out or perhaps pulling in to account for growth or decay at that place. So you get equation for divergence of u, and then this gets to equation, Poisson equation for p, which you can solve, and then you get interface motion. Okay, so that can be done, and one gets a nice, probably should have put a slide in, one gets a nice growing biofilm. It grows at a constant rate in time, because basically at some point you have a constant influx of material. So it's not exponential or anything like that. You just, it's transport limited. And you reach a point where you just can't transport anything more, and you get to grow at a constant rate. Okay, so that's very good. But then it turns out biofilms are not flat. We have a 1D model, which of course that can't capture things that aren't flat. At least not always. So it's a pity you can't see this too well, but this one you can see pretty well. This is an old picture from Paul Studley, who's very influential in a lot of this mechanics stuff. And this is a picture again from Yellowstone. These are little towers, tubes of growing cyanobacteria, or mostly cyanobacteria, or well, they're partly cyanobacteria. It's driven by cyanobacteria. Even just if all you want to do is growth, let alone all the rest of the mechanics that go into this, a 1D representation might not be sufficient. So in fact, if you check that, if you check a 1D biofilm, so you take a 1D biofilm and then give it a small perturbation in even 2 dimensions, you'll see that that perturbation grows into fingers. This is a fingering instability. And the idea is simple. It's just that the substrate is up here, and there's this limitation in transport, and the closer you are to that substrate, the easier that is, and the faster you grow. So if you get a bump, it tends to get larger. Okay. This is a computation, by the way, without shear flow. So there is other mechanics in here. If you have shear flow coming across this, it could, for example, erode things, and it would erode stuff at the top of these bumps faster than stuff at the bottom. So it would tend to oppose this. So I don't know what happens when you put in shear flow, actually. Okay. So really, it's sufficient to do this in 2D, but just for no particularly good reason, we did this in 3D. This is a movie by Eric Alphys. It's kind of old now, but it's kind of a nice movie anyways. So this is actually a multi-species, multi-substrate case, but it gets the idea across of these things growing. And here's actually a real movie of a sort. So this is stacked confocal images made on the computer into a movie. There's no time in here, but this is a real biofilm, Pseudomonas aeruginosa biofilm. So you can see it kind of looks similar. Also just a nice movie. I'll wait for it to end.
Is that grown under close-out conditions?
I don't know, but probably. Oops. Okay. Just again, as an aside, I wanted to go back to the for simple modeling purposes, we're looking at homogeneous biofilms, so the density of biomaterial is constant everywhere, but going back to this microcolony embedded system, we noticed this basically the same instability there, and I don't know if it's been noticed before. So this is an example where you just play around with the model and you come up with a new hypothesis that maybe you hadn't actually seen, and so here's a similar kind of instability, cousin of the other one. So we start. So we have a layer here, and on top we have oxygen coming in, and the bottom oxygen leaving. Oxygen level is 0, say. And we start with, we don't have a homogeneous density of biomaterial, but it's perturbed a little bit. So there's a sine wave or cosine wave, whatever it is here. A little bit more dense, I guess, in the red parts than in the blue parts. And then you let that go, and after a while, this is the density picture. So look at the top in the active layer. Again, you have alternating more dense, less dense, more dense. So here, notice the difference was 0.1 in whatever units we're using, and here the difference between peak and valley is 0.2 or 0.3. In other words, that difference is growing. There's an instability. So how is that caused? It's not like there's fingers going out here. There's nothing like that. But the idea is where you have more dense things, you use more oxygen, and that drives more flux. So you lower the oxygen level, that pulls in more flux. And so you get more oxygen than your neighbors, and you grow more. So by being more dense, you can hog oxygen a little bit, and so we tend to get an instability which goes away from homogeneous biofilm. If you look at a lot of these biofilm pictures, you do, if you see what you want to see, you can often see, it seems to me anyways, a sort of characteristic scale of density in there. So these, I should say, these computations in the one before were done by basically Andreas Aristotelis, who's a postdoc. Okay. So this is good. We have transport, we have growth, which is great, and now we want to keep on going. We want to look. So this is over relatively short times. We want to look over long times. We need something to balance the growth. So now we're in a bit of trouble because we don't really know how that works. So to reach a steady state or quasi-steady state, or just look over long times, things have to balance on average somehow. And so we need something to balance production. Not really much is known about this, at least as far as I know. There's another movie from James Connolly. He's a graduate student. He's about to finish. So you have biofilm up here, obviously, and stuff is coming off of this big honking chunks of stuff. And it's actually accumulating down here and forming something new, maybe. So what's happening up here, I don't know, but at least some of this is mechanical and probably pretty complicated in what it's doing. There may be other things going on here. There could be erosion, and there could be detachment, could be decay of some type, viruses or something. I just don't know. If anyone does, I'd be interested to know what it is. Another aside, actually, that's kind of interesting here. If you do have a situation like that where you have material being taken off the top of the biofilm by erosion or detachment or something like that, so that's a competitive pressure in a way on stuff that likes to grow at the top of the biofilm in the active layer, the most the most healthy stuff in some way in the biofilm, but it's the stuff that's being removed most likely from the biofilm from these mechanical effects. And so if you write down a model for this, even a 1D model, right away you see that there's an issue. If you just take stuff off the top of the biofilm, then you'll lose the stuff that likes to live at the top of the biofilm, and what survives is the stuff that likes to live down at the bottom of the biofilm, even though it may not really be very competitive at the top. So, but you can fix that by just allowing a little bit of downward mobility. You don't have to move very fast, but if you're at the top, at least the model predicts, or at least I would say, the model predicts that there should be some mobility typically in biofilms, downward mobility even against where you might want to be. And if you do that even a little bit, you can make a much more interesting and robust ecology up here. Anyways, let's move ahead. So, so we're talking about detachment. Unfortunately, or fortunately, this is a non-Newtonian issue. So I'm thinking of Newtonian fluid dynamics. I don't know who here is acquainted with that subject, but this is a non-Newtonian issue. So we're going to have to move on to other and more complicated things, and we do this reluctantly because we don't like to add things to their model if we don't have to. But here's the situation. If you want to know about how this works, you maybe have to know something about this. So here's a one-slide primer on viscoelasticity, and it goes like this. So biofilm, it's a mess of cells and polymers. So you can think of it. There's a bunch of tangled up polymers, sort of like in a rubber band. You can pull on it and they're kind of tangled together. If you let it go, they'll snap back. And that's like an elastic body. And this is the equation roughly for elastic body. Sigma is the stress tensor. It tells you the stress forces from deforming something. And that changes in time. This guy is called the rate of strain tensor. Basically, the stress is equal to the strain, or related to the strain in a simple way. That's Newtonian elasticity, if you will. But then what happens? So let's say we pull on this thing and we don't let go this time. We just keep pulling. So we have these polymers. They're loosely connected. If you keep pulling, they'll start slipping by each other, and then you'll get motion like a fluid. Well, I get ahead of myself a little bit. So what happens is that if you keep pulling, these entangled polymers pull by each other, and you lose the forces, you lose the stress. So the stress will decay, and the simplest thing to do is a linear decay like this, where lambda is a decay time. Now, so you have these two effects. You just basically add these together to get an equation like this. This is the equation for what's called a Maxwell fluid. And it has an interesting property to it. So suppose you make a sudden deformation of something, and so at early times the time derivatives will be large. So this term is large compared to that one, and what you see is that you see this equation basically. You see the thing act like an elastic body over early times. Now if you've made the sudden deformation and you've basically stopped, and now over longer times this term will become smaller compared to this one, and then you'll be left with this, and you may recognize that that's basically the constitutive law for Newtonian fluids. For a viscous Newtonian fluid, the stress is proportional to the rate of strain, to the friction, if you will. So over short times, it's like an elastic body. Over long times, it's like a viscous fluid. That's a viscoelastic fluid in one slide. There's an interesting parameter, eta times equals lambda. This lambda times that d. Well, you can actually see it from here. It's the fluid viscosity. Okay, so let me show you some movies. Biofilms are viscoelastic fluids, and this was an observation made by Paul Stooley back in the 90s. It's pretty small. Here's the elasticity. So here what we have is the biofilm, of course. Notice the lumpiness, by the way. We have a fluid coming across here, and he's turning that fluid on and off. And this movie is looping. So it looks kind of funny. Also, so for some reason, it seems like the flow should come this way, but it's going this way. Anyways, so you see clearly the elastic behavior of this thing. over short time scales. Here's another movie. It's not as clear maybe, but still convincing to me. So this is now a view from above the biofilm looking down on the biofilm with a flow going across. It's actually, you can't quite see it there, but it's quite fast flow. And you can see, he calls these ripples, these waves across here. Well, you can see waves in an elastic body, but these are actually, or at least Paul believes, are material waves. So it's actually material that's moving along here. It's not a wave that's going through the material, but the material is actually in the wave. In other words, there's flow downstream. This is like a fluid here. These are over long times. I forget what it is, but over hours or something, you see something like this. There's some other interesting things. There are some guys that don't move at all, so there's some heterogeneity in here, but that's a different story. Lost my... Okay. So what we did, fearless amateurs, led by Paul Studley, is we went out and we collected a bunch of biofilms, and here's one example. This is actually a natural biofilm, and we stuck them in a rheometer, parallel plate rheometer. So what this is, you have two plates, you put your sample in between, and then you apply a torque to the top plate. So you try to twist this thing with a certain amount of force. So here's the torque. We switch it. We apply a certain amount of force for a while, and then we actually drop down to 0 to see if this thing will snap back. And here's the data. Here's one piece of data. So you apply this force, and what you get is basically a sudden jump, like an elastic body would. And then you wait a while, and it starts deforming in a linear fashion, like a viscous fluid. So this is really a cartoon. This is a textbook picture of a linear viscoelastic fluid here. It's just perfect, actually. Then we release the torque, and there's a certain amount of snapback, but not all the way back to where it started from. So you've lost some of your memory here. And then there's a relaxation. So that's the one part that isn't like linear viscoelastic fluid, which would predict a larger relaxation than the one we got, and we still don't know why that is. This isn't just for one biofilm. We did this for a lot. I don't know if these are readable, but a bunch of lab ones. Here's Pseudomonas aeruginosa, PA01, I think that was mentioned. Some other ones, some natural ones as well. And we got the same kind of pictures all the time. Another interesting thing here is we plotted the viscosity of these guys versus the shear modulus, the elasticity, roughly speaking, and they all lie on this line. Now there's a huge range of elasticity and viscosity, enormous range, but the ratio is on this line. It turns out that this ratio is, roughly speaking, this time scale between elasticity and shear flow. So in all of these things, we basically got the same time, about 20 minutes. I was never sure if I really believed this result because we were amateurs, but it seems to have held up over the years. We don't know why. So there's a universal time, roughly universal time, maybe, for biofilms to go between elastic behavior, so they remember what happened to them and they want to go back, and then at a later time they say, just forget it, we're going to go with flow. It's about 20 minutes, and 20 minutes, of course, is kind of an interesting time biologically, so it's suggestive, but we don't really know what's going on. Okay, oops, I'm running low on time. That's all right. So all of this is leading to trying to deal with detachment. That's where we're trying to go anyways. Here's another movie. Another old movie, but it's still gets the point across. From Paul Studley again. So here we have a streamer from above, and this is the flow rate here. It's stretching out. Look how lumpy it is again. He changes the flow rate a bit. Now it's down a little bit, and then it'll go back up, and then it pops off. Okay. That's what we want to know. What is the mechanics in here that are doing this so we can get this right? Okay. Oh, so the we have viscoelasticity. By the way, I should have said. So I should have said on the last slide, you have this 20-minute time period, and one nice thing about that is that if you're looking at times over 20 minutes, basically longer than 20 minutes, you can, or roughly that amount of time, you can think of biofilms as fluids. Makes things a lot easier. Elastic bodies are difficult to deal with. They tend to, be elastic and spring around and go unstable and stuff like that. So anyways, we know the viscoelasticity, but how does that actually tell us? So that tells us how things deform, but how does that actually tell us how they split off? So what we did is we went in and we added a cohesive energy like this. So we have biomaterial volume fraction between 0 and 1, and the rest is water, say. And we say, well, there's a preferred mixture, say, of biomaterial and water. And if you're somewhere else, there's an energy that wants to push you down. Well, in principle, wants to push you. So bring in water or expel water or something like that. So the idea is this is what keeps a biofilm together. It wants to be at a certain volume fraction. Okay, so this gets a bit more technical, but for those of you who are interested, so here's the energy, and this will look familiar to some people. So we have this cohesive energy, and this is sort of like a surface tension energy added in there too. And then you have equations like this. These are Newton's law now, very complicated versions, where the biomaterial has this stress coming from this energy plus other terms. There's friction terms and other things that I didn't write down. If you play around, out of this pops the Cahn-Hilliard equation, by the way. Well, it's a bit of an awkward one. It has these coefficients which are degenerate at the ends. So it's hard to deal with, but I won't talk about that. Okay, so here's the kind of computation that we want to do. So let me explain this. So time is along this axis. So we start from here and we move forward in time this way. And this is biomaterial. And then there's a wall at x equals 0 and x equals 1. And the wall is actually moving apart. You can think of it as moving apart slowly. So we start off with a biofilm between the two walls, and they get stretched. So the biomaterial volume fraction goes down, it gets unhappy, and it snaps off the wall. This is a detachment event. Well, okay, so it's possibly a detachment event. It's still basically a slow linear type thing, and when you actually look at biofilms, it looks very violent when you see a detachment, and that's probably because we have all these polymers that are breaking and doing very nonlinear things. So we also tried this, and this was, again, really for fun, made it more than anything. So what we did, just as a demonstration, is we took a biofilm model where we discretized the biofilm into a bunch of points and connected them with springs, nonlinear springs that broke if you stretch them too much, sort of like polymer might do. And then there's an invisible wall up here that starts moving and drives a flow. This, by the way, it's periodic, so the stuff that comes out comes in the other side. And at least visually, I mean, this seems to capture things more, but we really don't know what's going on, how this compares to a real biofilm. Okay, so I'm going to have to think about finishing. Okay, so here's my long-time biofilm model. I don't know what it should be, so let mean, you can add, so you have, if you want to balance things, you have to add some sort of ad hoc mechanism, and there are various ways to do that, and various ways people do that. But whether, and you have to do that if you want an equilibrium, but whether you get something, how meaningful it is in the end, I don't know. I don't have the intuition to tell you. Okay, so I'm going to skip this basically, but I was also going to talk about, I was excessively optimistic. I was going to talk a little bit about chemistry as well. This is a picture again from James Connolly of mineralization forming, this is biofilm out here, mineralization forming on a glass plate due to pH chemistry. So I have quite a bit, but I'll skip it. Okay, so just to recap, biofilms, they are physical materials, they're polymeric fluids, they're chemically active, I didn't talk about that. They're living and growing and decaying and engaging in mechanics of various things often, and they're heterogeneous in all these properties and spatially dependent on all these properties, and they all have important consequences. And I just want to say, I tried to sneak this in at the bottom, omics are important, but they're not everything here. They don't, you need to somehow connect them to your physical environment to really figure out what's the right way to think about them. At least that would be my opinion. Okay, so I'll acknowledge various people. I'm sure I've left out some, but all these people are important for what I had to say. And thank you.
So we have time for a few questions.
Wondering if cell death is important, do you think, and how you could sort of incorporate?
Well, you tell me if it's important. It's not hard to incorporate it. So you have a growth term, and you just add a death term. In fact, in some of the simulations, I didn't mention that we do have that in there, but I don't know how important it is.
So in those kind of simulations, then do you see an effect on the structure of the biofilm due to including cell death?
Well, there's one in particular. I showed a fingering instability at the beginning. We put in cell death there just to keep this thing from growing out of the box. It did have some effect on the structure, but not dramatic effect. It could, but if you start thinking about mechanics, then you might think that, well, yeah, it might have an important effect there, but I don't know.
There's some experimental work suggesting that buckling on the surface of the biofilm is driven by cell death.
Yeah, so that I would think that might be a mechanical effect. I've seen some of that. Yeah, it's interesting.
Do you think that the detachment could be related to nutrient content more? Like, because with your growth equations, it seemed they were more related to the nutrient environment, whereas the detachment stuff was more mechanical. Have you thought about trying to incorporate?
So you could say detachment is sort of inherently a mechanical thing, but the material properties could certainly depend on nutrient level.
I'm just wondering if there's some theory that biofilms might migrate depending on nutrient content. So it might be more of a...
Yeah, so you're thinking this dispersal stuff?
Yeah.
Yeah, I don't know if I mentioned that or not, but you could certainly, if you get into a certain environment or a certain history, you could decide to change the mechanical properties in a fairly dramatic way and get something like that. But yeah, I just do the models. I don't know the answer to that question.
Just to pick up on what Orkin said with regard to this fingering property you looked at. So you framed it as a function of preferential nutrient access, like this ability for the biofilms to grow up.
Yeah.
But in addition to this buckling thing that Orkin mentioned, there's a whole constellation of biological theories out there to explain this, many of which have relatively little to do with preferential nutrient access. Now, I'm sure everybody would agree, perhaps even as a result of that model, that this nutrient property is playing a role in accelerating this. But there's definitely a line here between two camps. So I wonder if you would be inclined to accept that a physics model is fundamentally descriptive when the mechanism in question is controlled by a biological system.
No, physics is there. I mean, you can't. The laws of physics, you can't get around them. So if they're there, they're going to happen. Now, there could be other things. There could be biological control that are exerted on the physics to influence or stop it in some way. And there could be, for example, there could be situations where the physics doesn't dictate that you'll get fingering, but the biology does. So those are all possibilities, but I wouldn't say that physics is descriptive, no.
Other quick question? Quick one.
If I right understood, in your model of the active layer of biofilm, you're assuming that the source of oxygen and nutrition is coming from one side.
Yeah, yeah.
However, like in the nature, it's not uncommon when the biofilm is growing on substrate and the oxygen going from one direction and the nutrition from another. How do you think is it possible to model that, and how it might affect your model?
Yeah, that's yeah. For simplicity, I didn't really mention that, but that's certainly possible. Mathematically speaking, it's no problem doing that. You just change the boundary conditions from homogeneous to something else or whatever. It can affect things a lot. For example, this sort of fingering stuff. If you have things coming from both directions. If you have growth on the bottom, say, so the finger is driven by a growth layer at the top. If it's at the bottom, it's been a while, but my memory is you don't get the fingering in that case. It's stable. I could be wrong, but that's my memory is. So it could affect things a lot. Yeah, definitely.
In the rheometer, what state are the biofilms in before they go into the rheometer? I assume they're very mature biofilms, or are they grown in there? Or?
No. Well, the natural ones we just took samples and tried to get them back to the rheometers as quickly as we could.
Right. But they have no capability of growth within the rheometer.
No, well, that's pretty fast. The rheometry measurements are a few minutes or something. For the biology ones, I don't remember, but we were doing some of this colony method where you grow it up on grow biofilm on a filter and transfer it from agar plate to agar plate. So that's very convenient to just swap under the rheometer. I don't remember if we did all of them that way, but at least some of them.
Okay, let's thank the speaker again. Half an hour break.